Good afternoon. My name is Yvonne Pannekoek. I work at the Department of Medical Microbiology in Amsterdam, and today I'm going to tell you about the recent developments in the field of Chlamydiales. But before I start my talk, I would like to thank the team in Oxford for giving us the opportunity to work with Chlamydiales and using the database. Thank you very much for all your support during the last 12 years. And of course, I also want to thank Arie van Ende, who actually initiated this project in 2008. Okay, so what is new in the field of chlamydia? The number of species is expanding. We discover novel animal reservoirs, and in this way, the zoonotic potential is also expanding. And we use MLST and RMLST not only to study the population structure, but also for source tracing and for the identification of novel hosts. So what are chlamydia? Chlamydia are gram-negative obligate intracellular pathogens. They are characterized by a unique intracellular developmental cycle. And here you can see an infected HeLa cell. And in this vacuole, you can see the green replicating bacteria. They have a small genome. They share about 780 genes, and the DNA identity is ranging between 50 and 99%. And we are now able to make mutants, which is nice. So here is a phylogenetic tree, which is an old tree, and I will show you an update of this tree later on. But what I want to point out here is that there are three members that carry zoonotic potential, and this is Chlamydia felis, Chlamydia abortus. The placenta from sheep can be infected, heavily populated with bacteria. And this way, this bacteria can be transmitted to pregnant women, and this can result in preterm delivery. And of course, we have Chlamydia psittaci, causing psittacosis, which is a severe form of a pneumonia by humans when it's transmitted to humans, much more severe than the pneumonia caused by Chlamydia pneumoniae. And then, of course, we have Chlamydia trachomatis, which is a strictly human pathogen causing sexual transmitted disease and blinding trachoma. So when we started this work in 2008, we used like the DNA sequences of approximately 50 isolates, and 9 species were then present in the phylogenetic tree. And so this is the first phylogenetic tree that we constructed using the concatenated sequences of 7 housekeeping genes, the fragments of 7 housekeeping genes. And we were very happy with these results because a year later, this phylogenetic tree was published, and that was based on DNA sequences of 110 conserved proteins. And you can see that the grouping from the different species in the tree is the same as in our tree. So that was a very nice result, and we were happy with that. In the second paper that we published, we used more isolates, and they were isolated from a wide variety of animals. They are depicted here. And then we actually made a couple of very interesting observations. So when you look at a tree that is composed of different isolates from Chlamydia psittaci, you can see different things. The first thing, what we saw, is that the genotypes, so sequence type 24 here, is really associated with the host. So there is an association between genotype and a particular host. So sequence type 24 was only found in parrots and parakeets. Well, this is another sequence type, and this sequence type is only present by ducks. So that was the first interesting thing. Another interesting observation was that Chlamydia psittaci was not only found and isolated from birds, but also from a cow. So this was the first isolate that was found associated with a cow. And another thing that was interesting, if you look at the phylogenetic tree of the modern birds, then you can see here that the isolates, the turkeys and ducks, they group together closely, and the pigeons and the parrots are more distantly related in the tree of the birds. But if you look at the Chlamydia psittaci tree, so the isolates from Chlamydia psittaci, then you can see that isolates from turkeys group close to the isolates from the pigeons. and the isolates from the ducks group close to the isolates from the parrots. So that's different than what you see here, which actually means that the Chlamydia psittaci tree does not follow the phylogeny of their main hosts. And this is indicative for a host species jump that happened independently of the evolution of the birds. So now we are in 2020, and here we can see that many researchers that they submit their isolates from many different countries to the database, resulting in a database that is populated with almost 5,000 isolates. And we also have a genome collection. We now have more than 700 genomes that are annotated, present in the database. So this gives a really rich source of data. And many people use these data to generate the research and to write the papers. And this is one of the publications that came out from a PhD student in our lab in close cooperation with the group in Oxford. Okay, and I was talking about novel species. And my last couple of slides, I will show you more details about these novel species that are indicated here with the green dots. And I generated a new phylogenetic tree that was also recently published in a paper in Emerging of Infectious Diseases, in which we described a new, a novel species, Chlamydia avium, present at pigeons and parrots. And in this paper, we described that the potential, the pathogenic potential of Chlamydia avium in pigeons. I encircled the host of the novel species in green. So here you can see a snake, which is a new host. And apparently snakes are also colonized with chlamydia. We do not know the pathogenic potential yet. Here are 2 candidate strains and one new species, and here is another species that was isolated from snakes. Here you can see a species isolated from the ibis. Chlamydia gallinacea is present in chicken and turkeys, so poultry. And here we have Chlamydia species isolated from a red-shouldered hawk, so Chlamydia buteon. So they are all novel species. But there is more going on. Recently, a paper was published in which they isolated Chlamydia abortus strains, and these abortus strains were normally only found in mammals, but in this paper they described Chlamydia abortus strains which were characterized by a different sequence type than all the other abortus strains, and they were isolated from wild water birds. So that was a new development in the field. And there is another interesting development in the field. So this was described in Australia, a novel mammalian host, also representative of a novel zoonotic reservoir. And this was described in this paper, in which they showed an outbreak of psittacosis at the veterinary school, demonstrating a novel source of infection. And this is the source of the infection. This is the placenta from a mare that gave a preterm delivery of her foal. And this placenta turned out to be heavily colonized with Chlamydia psittaci and multi-locus sequence typing, which was done by Martina Jeloknik in the group van Adam Polkinghorn. PubMLST identified an avian-like Chlamydia psittaci strain, and that carried sequence type 24. So this also shows the importance of typing for source tracing, excuse me. And then we have a last fellow that is coming into our tree, and this is the Chlamydia caviae. Chlamydia caviae was already present in the tree, actually, but it was not known to be a zoonotic reservoir, the guinea pig. But in the Netherlands, we had a paper published in 2017 in which 3 cases are being described of patients that were heavily infected with zoonotic Chlamydia caviae, and they were hospitalized, and they were in the intensive care unit, and so they had to be treated with mechanical ventilation. So the message from this paper is that both veterinarians but also physicians should be aware of the zoonotic potential of Chlamydia caviae that can be carried in our pets. So I updated the tree a little bit, showing novel zoonotic reservoirs here and novel host species for Chlamydia abortus. And that brings me to my summary slide. The number of members of the genus Chlamydia is expanding. The number of host species of members of Chlamydia is expanding. And Chlamydia psittaci strains are not restricted to birds, but also found in mammals. Chlamydia abortus strains are not restricted to mammals, but also found in birds. And MLST of Chlamydia provides an excellent tool to study the population structure, to trace sources of transmission, and to identify zoonotic members. And this is my last slide. I want to thank the audience for their attention, and I would like to thank the group in Oxford for their support. Thank you very much.